Hello, fellow anatomists. Here we are, starting chapter 2, a little bit of backtracking, but I kind of like this order. So we are in week 5, module 5, cells, chapter 2. So cytology is the true term for the study of cells. I thought I'd start out again with what we don't know about cells, and that is cancers. The study of the cell is such an important study. And there's some things that we still haven't figured out. We still haven't figured out why it is that some cells will overproduce, why they go into mitosis and do it again and again and again and form a tumor. And it seems like such a basic piece of information that we're missing in this puzzle of cells. So let's start, thinking about that. This is a cancer, a picture of a liver full of neoplasms or tumors. Very scary. So this is, the classic case of cell division is exceeding cell death. And that balance of cells dying and cells being made is disrupted in a condition like this. So that we know this is caused by some mutation in the DNA, and then it starts to uncontrollably divide. So we're going to look at some terms that have to do with these cancers and with the words that are used in science and medicine. An agent that causes a mutation is a mutagen. So if it causes a mutation, for example, UV light, it's going to cause a mutation in your cells, it's a mutagen. An agent that induces cancer itself is a carcinogen. Like the molecules within cigarettes that have carcinogens in them, those molecules are able to induce cancer. An oncogene, however, is a mutated gene that promotes that self-growth. So a mutation's happened, now it's an oncogene, and it is that gene that is able to promote this cell growth. Then we have some very cool genes also, which are tumor suppressor genes, and they inhibit cell growth. They also promote DNA repair. This is so amazing in cells. That's why the whole study of cell biology is a course in itself, and we're giving it just a few little hours here. So we do have this tumor suppressor genes that are trying to always make sure that this growth isn't happening, and most probably they catch a lot of little small growths, but some get away. We know that tumors can kill. So I want to tell you a very interesting story about Henrietta Lacks, and many of you might know about this wonderful lady whose cells had this amazing capability of dividing and dividing and dividing, and therefore she died of cancer because of these cells. And at the time, they collected these cells from Henrietta, and they looked at them in the lab, and they were alarmed at how easily they were produced. And so these cells now went to all the cancer labs, and these cancer labs started to look at these cells in vitro, in a tube, right, or in a petri dish. And then this happened in the US, and then our science colleagues in Europe wanted some of these cells, and they were sent there, and it turns out they were sent all over the world. And those HeLa cells, even though she's long dead, have lived on. And it's very interesting YouTube for you to watch, and I've posted that in the other section. So these cells still live on, and they are in many labs, and people are doing research on them. And it did create some controversy in that she or her family was never asked for permission for using these cells. Thank goodness that's now been remediated, and now the family has a say in who gets the cells and what kind of research is done on them, which is interesting. So there's a wonderful book on it too, but if you see the video, you'll be referenced to it. So we need to understand these cells. You've already been drawing them. You know what a squamous cell is. It's one that's flattened. A cuboidal looks more like a cube. A columnar cell is taller than it is wide. And all these different shapes of cells. They come in so many different forms. Discoid, fusiform. I don't need you to memorize all these, like discoid that you didn't use in your drawings. So we say that the cell is a structural and functional unit of an organism. You can't get any smaller and still be alive. So it is truly the functional unit, the smallest you can get. They vary a lot. There's about 200 different kinds of cells. Most of them are tiny. Most of them are about 15 microns wide, which is 1/1000th of a millimeter. I've put the millimeter up here again. If you see one little guy between two of these little lines is a millimeter. And most cells are 1/1000th of that millimeter, so they are indeed very tiny. Some can be super long though. Like a nerve cell can be as long as, longer than a meter. Longer than 3 feet. So this is, the diversity truly is mind-boggling. There's some other good examples in nature, not in humans, but the nerve cell of a giant squid is the actual longest of an animal cell. An ostrich egg has the greatest volume. Look at that's a picture of an ostrich egg. They're so beautiful. And this is the largest single-celled cell. And it's, what was the name? I'll have to get back to you on it. Like, it starts with a V, and it's kind of it's aquatic, lives in the water, and this is one of those largest single-celled organisms that's a single cell. So we want to look at the two fundamental subdivisions of a cell now, and back to humans. The cell membrane, or the plasma membrane, is also synonymous, and everything that's inside. So one is the membrane that you've already been drawing, and everything else that's inside, which is the cytoplasm. Now the fluid, this is confusing because there's so many words for the same thing. So ICF is intracellular fluid. It's the fluid that's inside the cell, right? It's in here. All this is intracellular fluid. That's good. However, it has other names. So the extracellular fluid is everything that is out here. And it can also be called tissue fluid, or it can be called interstitial fluid. So these three, extracellular, tissue, or interstitial, are all words for the same thing. It's for all the fluid here that surrounds the cell. So let me back up. The intracellular fluid really just only has one name. It's either cell fluid or intracellular fluid. Okay, when we show you a cell like this, I always laugh and say that this is Joe's cell, because there is so much variability between cell to cell that to think that you could just draw one and put all the structures on it and make it a real cell is not true. So this is just sort of Joe's cell, where you're trying to pile everything in on it, and then you have a description of the structures and what it could potentially look like. So I'm going to give you a recipe here if you want to build a good cell membrane, right? You're going to build a cell and you have to start with the membrane. And how would you actually start building that outer membrane? So you need ingredients, and the ingredients would be 2 layers of lipid. So we're going to call it a bilayer because it's 2 layers of fat, 2 layers of lipid. You're going to need a handful of proteins. And you're going to need a pinch of cholesterol. And you're going to need a dash of sugar. And these are going to assemble in a double layer. So let's see those in more detail. So 2 layers of lipid. we have this outside region of the cell and all these little balls here and all these little balls here, but the lipid part is this. I'm going to put an L there. That's the lipid part here. And it's 2 layers because you see balls and balls, right? Handful of proteins. Here's a protein. Here's another protein, right, in green. They're kind of stuck in that double layer. Cholesterol, I'm going to draw it in. It's going to be sort of in regions like this. Within this fatty layer, I'm going to add some cholesterol here. There we go. That's cholesterol. And just a pinch of it. And then a dash of sugar. Well, these chains, if I change my color of my pen, these, I'm going to circle it there. Carbohydrate chain is the one that's protruding here from the protein. The protein is green, but this part here is carbohydrate chain. You'll see that sometimes just coming off those membranes. So that's the sugar. Okay, so this membrane is arranged in this double layer, and I want to show you more detail about it. So I'm going to stop this video here, and we're going to start the next video with this diagram and talk a lot about what's making up this membrane. Thanks for coming.